Yeah.
Very good.
Because English wasn't hard enough.
Okay, so here we are. So welcome and thank you for joining us for today's session on frequently asked questions from the International Myeloma Society annual meeting. I'm Mary DeRome, Senior Director of Medical Communications and Education at the Multiple Myeloma Research Foundation. Today, I'm joined by Dr. Jesus Berdeja and Nick Barkemeyer from Tennessee Oncology in Nashville, Tennessee. We've invited them here today to answer some of the frequently asked questions we receive from patients and caregivers about the data presented at this year's IMS meeting, International Myeloma Society. So let's jump in. We're going to start talking about minimal residual disease. So there were several presentations at IMS focused on the importance of minimal residual disease, or MRD negativity, including using peripheral blood to measure MRD. Dr. Berdeja, welcome to the show. And can you tell us how the use of MRD is evolving in myeloma, and how close are we to being able to measure MRD with a simple blood draw instead of a bone marrow biopsy?
Well, first of all, thank you for having me again. So it's always a pleasure to talk with the MMRF audience. It's always very savvy, I might say. So MRD, just to kind of back up a little bit, obviously is a deeper way of measuring a response. So when you get a complete response, we know that there's often cells that are residual, and MRD is just a way to sort of kind of get a deeper read into that response. So by definition, the deeper remission you are, the longer it takes for your disease to potentially come back. So the traditional way of assessing MRD is with bone marrow biopsy, and either with flow or with this other procedure called NGS, or next generation sequencing. And that's the traditional MRD that people probably have come to see and know from most clinical trials, but that does require bone marrow biopsy. But there are other things that also can tell us how deep the remission is, especially in the patients that have disease outside of the bone marrow, for example. Imaging is very important to assess minimal residual disease with things like PET scans and MRIs. And then in patients who have extramedullary disease outside the bone marrow, sometimes bone marrow MRD is not indicative of the true response. And so something like a blood test that can actually assess cells everywhere, not just in the bone marrow, is very attractive for that reason. And luckily, there are some new tools that we have at our disposal that are starting to yield excellent results. I think from the data that was presented at IMS, we saw a lot of the traditional bone marrow MRD being used. Can it help us guide therapy, for example, the duration of maintenance, for example, which is a question I get asked a lot by my patients. Can I come off my Revlimid, for example? So there were some really nice studies looking at patients who achieve MRD negativity, but not just one time point, but actually several time points. In one study, it was 3 time points, where basically we call that sustained MRD negativity. So in a patient that has a complete response, who is MRD negative and potentially PET negative, those patients, after 3 years of Revlimid, had their lenalidomide stopped. And the majority of those patients were able to stay in response. So that's actually encouraging, that perhaps at some point we could use MRD to help guide how long our treatments continue. I will caution, though, that there are some studies looking at patients who have sort of higher risk disease where that has been tried, and we know that in patients, especially with ultra high risk disease, meaning you have more than one traditional high risk feature, stopping therapy led to quickly progression of disease. So again, it's, as in myeloma, as in everything else in myeloma, is one size does not fit all. So it's important to make sure you kind of treat the data with caution. In terms of the blood test, which is what Mary actually asked, Mary knows I tend to ramble, so Mary, please stop me if I.
It's all good information.
There's several things that are ongoing right now, but one of the tests that we're all very excited about is called mass spectrometry. And that is actually a test or a procedure or process that's actually designed to try and replace the traditional SPEP and UPEP, which you all have come to love. And so we know that with that technology, we can assess a much deeper, or is much more sensitive. So you can, if it's truly negative, we know that the patient has a much deeper remission. And so that is actually is used for that reason, but also now it's being looked at as a potential can achieving a complete response by mass spectrometry actually equate MRD negativity by bone marrow? And the short answer to that is that we don't know. But the longer answer is that, and the Spanish have sort of kind of led this effort, is that it does seem to be just as powerful as MRD in the bone marrow, but they don't seem to be equivalent, meaning that they're complementary. And so actually using both perhaps would be better. But again, that data, this is all retrospective data. So as usual, we need studies to help us determine how to best use it.
Sure. So yeah, I mean, this is something that patients are eagerly awaiting, because I don't think there's any patients who really enjoy having a bone marrow biopsy. So Nick, are there some patients for whom a blood draw instead of a bone marrow biopsy to measure MRD would make a big difference? For example, do you have patients who are basically unable or unwilling to undergo a bone marrow biopsy? Certainly on this format, we've had some patients who have come on who have said that bone marrow biopsies can be very painful. And I think that the way that their care teams try to help mitigate that pain differs from center to center.
Yeah. So first of all, thank you for having me here today to talk about this. Obviously, myeloma is a topic that I love to talk about. So yeah, I mean, I think there really are patients that would much more rather have a blood test than a bone marrow biopsy. And I think that number would probably be 100%, because who willingly wants to go through a bone marrow biopsy? But from a practical standpoint, I think that, the ability to do that via blood work as opposed to biopsy would benefit them in the sense that, maybe the patient is very frail. Maybe they have difficulty dealing with, if the patient's going to be sedated for the bone marrow biopsy procedure, maybe they've had difficulty with the anesthesia in the past.
Right.
Maybe they're on a blood thinner. They have to hold their blood thinner for, so many hours prior to the procedure. And as Dr. Berdeja mentioned too, if you could have the ability to check for disease outside of the marrow as well, broadening that scope is actually going to be more beneficial to the person than just doing a bone marrow biopsy.
Right. So more to come on that, and looking forward to seeing some more interesting data from mass spec and MRD measurement in blood biopsy instead of bone biopsy. So let's pivot a little bit to the data that was presented for different drug combinations for both newly diagnosed and relapsed refractory myeloma. There were a couple of presentations on Venetoclax in combination with Kyprolis and dexamethasone, specifically in patients with the 1114 chromosomal translocation. Dr. Berdeja, it seems like we've been waiting forever for this drug to be approved in myeloma. So can you remind our audience what Venetoclax is and why it's being studied only in this specific patient population? And when is this drug going to be approved for myeloma patients, even though many myeloma patients already use it who have 1114 in compassionate use settings?
Correct. Well, don't say that too loudly. So myeloma, as you all know, one patient's myeloma is different from another's, but in about 20% of patients with myeloma, there is a translocation of chromosomes 11 and 14, meaning chromosome 11 and 14 broke off, came together, formed a new chromosome, and that is considered the 11-14 translocation. We know that patients who have that translocation, their myeloma cells are specifically dependent on this protein called BCL2, and BCL2 allows those cells to survive. And Venetoclax is a drug that was approved actually for a different type of cancer, a sort of cousin, like specifically CLL and now also AML, where we know that BCL2 is an important component of the survival of those cells. And so it makes sense that in myeloma it should work, and it does. It works very well, but only in the subset of patients with translocation 1114. The reason this drug is not approved is that when the original study done to hopefully take it to approval to the FDA was done, it was done in all patients, not just the 1114 translocation.
I remember those days.
That was called the Bellini study. And so that was the Venetoclax with Velcade and dexamethasone compared to Velcade and dexamethasone. And although the study met its primary endpoint of progression-free survival, it actually showed a worse overall survival for the patients on the Venetoclax arm, mostly because of increased toxicity. But when you look at the patients, the subset of patients with just the 11-14 translocation, those patients actually benefited both with progression-free survival and overall survival, thus proving the point that that's what you need. So actually at the IMS this year, I think that probably the most important presentation was actually that we're all waiting for was the CANOVA study that was presented. And that study looked specifically at patients with the 11-14 translocation, and they were randomized to receive either Venetoclax with dexamethasone alone as a doublet versus pomalidomide dexamethasone in patients who had prior PI and IMiDs. And although the Venetoclax arm outperformed the other arm, the progression-free survival was 9.9 months compared to 5.8. The overall response rate was double. The time to progression to next treatment was almost, I believe it was double as well. The progression-free survival benefit did not meet statistical significance, which was the primary endpoint of the study. And so it is considered a negative study. So we'll see how the FDA views this in light of the information I just told you. So it's very frustrating. But what's interesting is actually at the same meeting, we had also the data presented with Venetoclax in addition to Kyprolis dexamethasone, and there we're seeing some very impressive results compared to just Kyprolis dex, with overall response rates in the mid-90s and with progression-free survival that is approaching almost 3 years, very similar to some of the more powerful combinations we have. And so in my mind, I think that's what it's required. Remember that the 11-14 translocation, although present in 20% of patients, in any individual patient, it's not present in all cells necessarily, meaning that if you just use the target by itself, you may only affect those cells, but then the others will actually cause progression. So I feel it needs to be done in combination. I think that's how we're going to get it approved. So hopefully it will be approved because it is a very powerful drug. It's just hasn't been tested appropriately in my mind.
Yeah. Again. I don't know. Eventually we'll get this right, I think. It's going to take some time.
Right.
So also there was some data on some other triplet and quadruplet combinations in both high-risk newly diagnosed myeloma and relapsed refractory patient populations. So what were some of the take-home messages from some of those studies?
Yeah, so, again, the high-risk patient population is a very difficult population. And usually high risk, just to kind of back up a little bit, means that the patient's myeloma cells have these high risk mutations, in particular 414 translocation, 1416 translocations, deletions of 17p, amplification or extra copies of chromosomes 1q, and having a disease outside of the bone marrow, extramedullary disease, truly out of the bone, etc. So those are considered high-risk features. So in that patient population, it's always the problem again is we get responses, but the responses don't last very long. So we had some updates at the IMS from two large European trials, one from the Germans and one from the French. The Germans is called the CONCEPT trial, and they are looking at patients with just high-risk disease. And actually, from the very beginning, newly diagnosed, they're using what we consider the four most powerful drugs we have right now. So they're using an anti-CD38 antibody. In this case, they're using isatuximab, Kyprolis, Revlimid, and dexamethasone as an induction. And then if patient goes to transplant, because not all patients go to transplant, they got transplanted, and then they get several more cycles of the 4-drug combination, and then they continue maintenance with 3 drugs: the anti-CD38 antibody, Kyprolis, and Revlimid. And so a very aggressive approach. And basically what they're showing is they can induce deep remissions, and those remissions seem to be lasting much longer than what we consider historically for this patient population. So more to come on that. I think the following these patients longer will really help us determine how much better this is potentially than our standard. The French are doing a similar thing, but instead of isatuximab, they're using daratumumab. But one of the difference is that they're not giving the maintenance with those three drugs. They're only doing maintenance with Darzalex and Revlimid. And so I think this will help us sort of be looking at both of these studies to determine how important is the addition of the proteasome inhibitor as maintenance as well. But more to come. And again, that early, it looks very good. And then the same in the relapse refractory setting. So, I think when disease comes back, some patients already have some high-risk features, but those can also be acquired. Each time the disease comes back, you can get high-risk features. And patients with high-risk features tend to do worse with any particular treatment. So there's a lot of work as well. But at least we're seeing, again, data with similar combinations that we just talked about. So we had the final overall survival analysis of the IKEMA trial, which is isatuximab, Kyprolis, and dexamethasone versus Kyprolis, dexamethasone in patients who had 1 to 3 prior lines of therapy. And I mean, that's probably the best progression-free survival we've seen, still about 3 years. For the triplet combination, and the overall survival, even after almost 5 years of follow-up, has not been reached. And so when they looked at patients with high-risk disease, those patients also benefited from the triplet. So I think that basically the moral of the story is the more the better for high-risk patients, and also keeping the treatment ongoing, unfortunately, is important.
Yeah, so it's interesting. We had a podcast that we did with patients who were high risk, and 2 out of the 3 patients were actually having triplet maintenance therapies, right? And they were actually doing okay on them, So, Nick, let's talk a little bit about managing these patients with high risk myeloma. So do some of these emerging combinations provide you with more options for managing this population? And are there more toxicities you have to worry about?
Well, I mean, yeah, I mean, all these therapies that are emerging are definitely the more therapies, the better, as Dr. Berdeja said, for these patients. So I think the key thing that was already mentioned was, yes, keeping these patients on therapy and having those patients understand why they need to go on therapy compared to other myeloma patients. So, I get a lot of cases where somebody will come in and say, oh, well, my friend has multiple myeloma, but they're doing this, this, and this. And then I have to explain to them, yeah, well, that's because not every myeloma is the same.
Right.
And so particularly with the high risk group, I try to tell them, we're going to try and keep you on an IMiD. We're going to try and keep you on a PI, and we're going to, maybe incorporate a anti-CD38. And so it will be interesting, like you said, to see some of these studies head to head, and hopefully that will kind of show the importance of, having the PI involved, as opposed to just, the anti-CD38 and the IMiD. Mm-hmm. So as far as toxicities go, I mean, obviously anytime you're on a multiple drug therapy regimen, there's always the increased risk for more toxicity. But what I'm seeing is that a lot of the patients with these kind of newer therapies that have been come up the past 5 years, they tend to do a lot better than, say, those that had high risk features 10 years ago or so.
Sure. Mm-hmm. Mm-hmm. Yeah, I mean, things have come a long way in 10 years, right? So, which is a great thing for patients. So we've had so many drug approvals in that time. So things are doing much better. So, okay, so shifting gears to some of the newer and emerging classes.
Can I just add something that makes sense? I think it's important. But the other thing is also, I think we've seen the importance of the multi-drug agents continuation. So adjusting doses for toxicity is important to try and maintain as opposed to stopping one. To try and maintain all of them going is important. So I just wanted to add that, because I'm sure patients will have questions about.
Sure. Yeah, makes sense. Okay, great. So, new and emerging classes of agents. So we saw several presentations on CAR T-cell therapies, and it looks like companies are really trying to speed up the manufacturing process and using imaging to predict relapse. We also saw data on the use of CAR T therapies earlier, that is in patients who've only had 1 to 3 prior lines of therapy. And right now they're only approved in patients who have had 4 or more, right? And last but not least, there was some phase 2 data on a new CAR T cell target, GPRC5D, which is actually an approved bispecific antibody target. And that was also presented. So Dr. Berdeja, what are some of the major takeaways from IMS regarding CAR T cell therapy for treating multiple myeloma?
Yeah, so I mean, I think we saw a lot of updates from what we've seen before, but I think that is you are correct. So in terms of one of the downsides of CAR T was many downsides. There's a lot of great things about CAR T, but one of the downsides of the autologous CAR T, meaning CAR T is using your own T cells, is that there's a requirement for collecting the T cells, and then you have to manufacture the CAR T before it comes back, and that usually on average is taking about 4 to 6 weeks.
Mhm.
And so during that time, myeloma can progress, and so people may need chemotherapy. And so we always trying to figure out how can we make this better. Obviously, the ideal would be an allogeneic CAR T, which is basically a CAR T that's off the shelf. But so far, those have not been as effective as the autologous CAR Ts yet. So in terms of trying to minimize that, we did see 2 presentations at IMS with new sort of faster manufacturing with sexy names like T-Charge and Insta-CAR Ts. So so these CAR Ts can be manufactured in 1 to 2 days, which is amazing. Now the problem is, because these are cell products, they still require that you do all this infectious disease testing to make sure that they're not contaminated, right?
Yeah, it's actually really that sort of QA that you're doing on these products that takes the longer time, right?
Correct. And if you're the patient, you want that, right? You do not want to get a product that's contaminated. It's going to be receptive. So unfortunately, that still adds time. So the fastest turnaround, even on the clinical trials, is really still about 2 weeks. But if it's 2 weeks versus 6 weeks, I still think that's a significant improvement. And so I think that will make a big difference for some of our patients. If anyone's heard me give talks about bispecifics with this CAR T, sometimes that's the patient that often goes to bispecific, is the patient that's progressing very quickly. They can't wait for that CAR T. Well, making these faster may open that up for some more patients.
Yeah.
But in terms of the earlier use, like you said, I think that's actually the most exciting thing that's happening right now. So we saw some updates on the CARTITUDE-4 trial, which is basically using, in patients with 1 to 3 prior lines of therapy, is giving CARVYKTI, which is a BCMA autologous CAR T that's now approved for 4+ lines of therapy, as Mary said, versus standard of care. And most patients got DARZALEX, POMDEX, and I believe Velcade-Dex. And so we saw the data earlier, and we know that the CAR T outperforms the standard of care, and so it's looking very promising. So basically, it's telling us that it's better than any other option you have. So that's the good news. This data is being looked at by the FDA as we speak. So we're all hoping that within the next year that we will have the availability to use CAR Ts as early as second line, but hopefully earlier than waiting for 4 lines of therapy. One of the things we saw here at IMS that was new was they actually looked at different sort of patient groups to see if anybody benefited more than others, because unfortunately high-risk features are still important even for CAR T and bispecifics. And so the good news is, at least on the CARTITUDE-4 trial, patients with extramedullary disease, with high stage 3 disease, high-risk cytogenetics, all benefited from the CAR T and was superior to standard of care. Now, we'll have to wait and see, but usually the benefit's not as much in the patients without these high-risk features. But nonetheless, it kind of helps us, or at least reassure us that we can still choose this treatment over another, even if they have these high-risk features. And then finally, I think you mentioned the GPRC5D CAR T. We actually saw 2 presented at IMS, and again, these were updates. One is a Chinese company that's looking at GPRC5D, and they presented their update, and then the other one is actually a BMS product that's been presented as well, that also was updated at this meeting, and both are looking quite impressive. So the GPRC5D target we know is a good one. As Mary said, we have a bispecific that's approved for that. But the CAR T is just as efficacious. We're seeing some very high response rates in the 80 to 90% response range. And the important thing is that a lot of the patients on these trials actually had prior BCMA therapies, and the efficacy was just as good, or seems to be just as good. So it tells us that we can use a similar therapy with an alternate target. And I think, especially with CAR Ts, I've had patients, and Nick can tell you this, who come who've had a BCMA CAR T, had an excellent response, and come and demand a second CAR T, and we put them on the GPRC5D CAR T and have had amazing responses. So hopefully that's what this will translate to. So more options, the better, as usual.
Yeah. One of the things that I thought was so amazing was they had, I think that it was at this meeting, they had a presentation on giving patients like a much lower dose of CAR T, right? Just many fewer cells, and then basically letting the cells grow and expand in the patient instead of growing it and expanding it in the dish before you give it to the patient, right? Which will then, shorten the manufacturing time. And the results of that I thought were pretty interesting, right? So.
Yeah. So actually, no, they bring up a good point. So that's part of the way they're decreasing this manufacturing time is called in vivo expansion instead of in vitro expansion. Most CAR Ts are expanded in the petri dish, and then they're given back to the patient where they expand in vivo as well. But what we're finding out is when the cells expand in the petri dish, they actually become activated. And so oftentimes what happens is then you give them to the patient, and those CAR Ts already can be exhausted,
Retired,
and or cause more toxicity. So the idea of sort of allowing for the expansion to happen in the patient themselves is what's driving a lot of this, and at least the data early on looks like these may be more effective cells.
Yeah, it was a better product, actually, is how they sort of described it, right? So, which I thought was super interesting. So, now that potentially CAR T will be approved for a new indication where it'll be available to patients who are in earlier lines, that is probably going to affect availability of CAR T, which has never been that easy in the first place, right? So Nick, from a practical perspective, has the process of getting patients access to CAR T therapy gotten any easier? And what will faster manufacturing mean to some of these myeloma patients?
Oh, well, access to CAR T has gotten much better now that, we've gotten commercial approval through the FDA for Carvykti and Abecma compared to, just having a clinical trial option a few years ago. Obviously, faster manufacturing can mean life and death to some patients. Unfortunately, there are those, as we talked about, high-risk patients that can progress very quickly. And if you have to sit around and wait, weeks and weeks on end before you could get the product, that could potentially be the difference between life and death. So it's really going to make an impact if manufacturing can be sped up, as Dr. Berdeja alluded to. I mean, 2 weeks would be, at least a 50% reduction in some cases for people. So, eventually, like he said, hopefully one day we will get to an allogenic CAR T where it can be just off the shelf. But, in the meantime, if autologous is what we have to do, but we can make it faster, then I think that's just going to mean better outcomes for people.
Agree. So there was also several findings presented for bispecific antibodies, including their role in treating high-risk myeloma, causes of drug resistance and strategies to overcome that resistance, as well as data on a new GPRC5D bispecific agent and an update on Tecvayli. So Dr. Berdeja, what can you tell patients about these recent findings for bispecifics?
Yeah, so I think that's the other pillar, right? So it seems like every meeting we go to, it's everything myeloma, and then about half the meeting is about CAR Ts or bispecifics.
Right, exactly.
So the good news is we have 3 bispecifics that are FDA approved in the United States, right? So 2 against BCMA, teclistamab and elranatamab, and then 1 against GPRC5D, which is the target we just talked about with the new CAR T as well, and that's called talquetamab. Or Talvey, I think. So it's Talvey. Thank you. See, I only know the generic names. So that tells you how encouraging this data is, and the data does look fantastic. But obviously, we're all struggling with how do we give these drugs, and they have the exact same indication as CAR Ts. So now we have 2 CAR Ts and 3 bispecifics with the same indication. And the question is, can we give them? Is it better to give one versus the other? Which one first? Which one second?
Sequencing is a huge question.
Sequencing is a big question, and so a lot of the data that was presented is really being geared towards that. So, we speculated over why patients stopped responding to both CAR Ts and bispecifics, but we really don't know the reasons behind why someone's disease becomes resistant. But more recently, we've seen some publications and now some data presented that tells us that the mechanisms of resistance may be very different between the CAR Ts and the bispecifics. For example, after CAR T, when a patient's disease progresses, it is rare that they lose the target. So, for example, if you get a BCMA CAR T, chances are at time of relapse, you will still express BCMA. And what we're finding out more is that patients who are getting bispecifics and then their disease progresses, we're seeing a much higher rate of negative antigen escape, meaning that the myeloma has lost that target, which would tell you that if that's the case, then going in with against the same target is unlikely to be of benefit as opposed to a CAR T. And we've actually seen that with some of the data that is now being presented, both from clinical studies and from the real world, where the sequence does matter, where patients who've had a prior CAR T seem to respond very well to a bispecific against the same target, but the reverse is not the case. And so I think from that standpoint, I think it makes sense that we pay attention to those kind of details, because I think that will be important. Changing the target, obviously, it's another strategy, and so that's why it's so nice that we now have GPRC5D and with talquetamab approved, but also we saw some data presented for forantinib, which is another GPRC5D bispecific that is being developed that is also looking very active. So that's important. We talked about the GPRC5D CAR T. So again, we will have options against GPRC5D, but also not presented at this meeting, but presented in past meetings. We know we have another bispecific that goes against a different target called FcRH5, and that's called cevostamab. And so again, having multiple targets is important. We've also seen data in other meetings where combining bispecifics against multiple targets may be beneficial. And so that actually is we're starting to see that. And so can we prevent this antigen escape by approaching against, by hitting 2 different or 3 or more targets? And now with all these, we have that possibility, similar to what we do with the PIs and the IMiDs, right? We never give them by themselves. We always give them combinations. So why not these? So again, I think that's all the data that's going to be forthcoming. It's going to be combinations and multi-targeted. But we're also starting to see that maybe you don't have to give multiple drugs. There's actually now studies with CAR Ts and bispecifics that are looking at multi-targeted bispecific and CAR Ts. And with one drug, you can hit multiple targets. So again, I think that's just a proof of concept, and that's what the data with the bispecifics really is showing us, is that they're effective, but how do we use them best?
Yeah. And I mean, dosing is a big issue too. I mean, we had a webinar last week on non-BCMA targeted bispecific antibodies, and we had Dr. Ajay Chari and also Dr. Suzanne Trudell on there talking. It was a great combination, and they had amazing things to say about about Talvey and about cevostamab, which is still in clinical trials. I was especially impressed by the cevostamab data, particularly the fact that many of the patients who were on that study were only dosed for 1 year, right? So they had 17 cycles of 3 weeks, and then they stopped treatment. And every patient who had reached a stringent complete response, by the time they stopped the therapy, still many of them had still not relapsed, and it's been 2 years since they were treated. So they were basically on drug holiday for some of them as long as 2 years, and they had no relapse, which I was not aware of. So it was very interesting data. So any of the patients out there who are interested in looking at that, just make sure you go to our website and check out some of our resources, and you can see a recording of that webinar that's already posted and see some of that really interesting data. But so also, speaking of other interesting data, Nick, we heard about some of the nail and skin side effects with the GPRC5D targeted agents. And we recently heard from Dr. Chari on this webinar that these effects may be correlated with good activity of the bispecific. And it was really interesting because we had a patient on the webinar too, who was also taking Talvey, and he had been really plagued with some of these side effects. And he was so thrilled to hear Dr. Chari say that it probably means the drug's going to work for you, right? So, which is good news for patients, right? So how do you prepare patients, Nick, to anticipate these side effects? And I mean, they showed pictures of some of these patients with their nails and their rashes and things like that. It didn't really look that pleasant.
No, it's not typically pleasant. So, I mean, obviously up front before they even get the drug, we kind of a chat with, you're probably going to experience some side effects involving your taste buds, your skin, your fingernails, potentially just one of the three, potentially just all three. Potentially it lasts, a couple weeks and then it'll kind of fade out or plateau, or this could be something that lasts for months. But me just telling them that isn't necessarily the best thing to do. So we've also, if both parties are amenable, we've had patients talk to each other and kind of share their experiences to let them know what possibly could be coming down the pipe for them. But I think that if a patient does have those side effects, first of all, they don't like them, but they do take solace in the fact that if it is working against their disease, it's something that they can manage and deal with. Right. But as far as preparing them, having them talk to other patients, explaining it to them up front, teaching them what to look for, some strategies to kind of help mitigate the side effects, whether it's, hard candy to try and increase, saliva to kind of help with the taste buds, or, there's lots of things out there to kind of help keep the fingernails from becoming so brittle. It's just about preparing them to have a plan of action before, those issues arise, because they know that one of them is going to arise. So,
yeah. I highly recommend a lovely manicure. So,
yeah, I wish there's a lot of our female patients that wish they could go to the get a mani pedi, but they're like, yeah, they just look so disgusting. I think I'll pass
right?
this week.
All right. So, oh, sorry.
Can I jump on something you both said?
Please.
First of all, Nick didn't tell you about, because we had this Vosma study open as well. And how long has our patient been off?
It's been over 2 years, 2.5 years. We saw her a couple weeks ago.
Yeah. So she's getting close to 3. So that's, and I think that brings up a good point, that a lot of the research now is also looking at, because it's not just with cevostamab, so it's not planned, but there's some patients on some of the other clinical trials with the bispecifics that come off for whatever reason, toxicity, or they get an infection, which is something we talked about. Infections are a big problem. Who actually have achieved a response, they come off and they stay in remission for a very long time. So the truth is, we're all hoping that this means that we could do limited duration of therapy for these bispecifics, and that and then preemptively restart, or hopefully it's just, you just have to give for 6 months and you're done after you reach a certain remission. Those are the kind of questions that we still have, because these toxicities that we're talking about do not always go away until you stop the therapy. And so we talked about the GPRC5D toxicities, but the infection toxicity is the most important. And so that risk of infection continues indefinitely while the person's in the bispecific. So I think if we could get to shorter durations of treatment, I think that would be optimal in my opinion.
Agree. Okay, so finally, we saw a few presentations on a new class of drugs called the CELMoDs. So Dr. Berdeja, how do these CELMoDs, like iberdomide and mezigdomide, or MEZI, as Dr. Richardson likes to refer to it as, what do we learn about its use in both newly diagnosed and relapsed refractory patients, and how soon might this be a treatment option?
Yeah, so these are exciting drugs. So these are in the same class as the IMiDs, which we know very well, thalidomide, lenalidomide, and pomalidomide. But when Revlimid, when Pomalyst were first came, and definitely Thalidomide, we first found out that they worked for myeloma. We had no idea how. We just knew they worked. And so then later on, these brilliant scientists realized that it was by inhibiting cereblon, which led to degradation of these important proteins for the survival of the myeloma. And so the CELMoDs now are drugs that have been developed after this knowledge. And so now these drugs are specifically directed to inhibit cereblon. And so they're much cleaner and much more powerful in that sense. And that's why they're no longer called IMiDs, now they're called CELMoDs, but similar class. So the CELMoDs, the one that's most advanced is called iberdomide, and that's we're hoping is getting close to approval. But that was actually presented here in combination, even in the patients who were previously untreated, in combination with Velcade and dexamethasone, showing that you can combine it as well as you can Revlimid or Pomalyst with our usual classes. And what we're seeing is that they unfortunately they do have issues with low blood counts, and that's actually by definition, because cereblon and Ikaros iolos are important for neutrophil recovery. So it's not unusual that we would expect that. But maybe Nick can eventually tell us that I think we're seeing less toxicity, and that's what this trial seem to indicate, is that they seem to be have less off-target effects. The other drug, and I think Richardson calls it Messi, like the soccer player. Mezigdomide is an even more powerful CELMoD that is also being looked at in combination, and that one seems to have more toxicity from a standpoint of the cell counts. And so because it is more powerful, I think we would expect that. But what's encouraging about mezigdomide is that when you look at patients with heart diseases, like subset of patients with extramedullary disease, meaning tumors outside of the bones and bone marrow, which often are very refractory to most treatments, mezigdomide seems to be very effective against that subset of patients. And so that actually would be quite impressive and nice if we had a drug that actually can overcome that kind of patient population. So yeah, these are looking fantastic. There are clinical trials ongoing, not only to look at combinations, trying to kind of prove that, to the FDA that they should be approved, but eventually, being looked at even as early as maintenance therapy after transplant instead of Revlimid, for example. There's a trial ongoing right now looking at a head-to-head comparison between Revlimid and iberdomide. So more to come from these drugs, but definitely these will probably sort of kind of replace potentially even some of our current IMiDs with more specific and more powerful drugs.
Yeah, that would be interesting. So we talked a little bit about the neutropenia that these drugs cause. So Nick, are there any other side effects that people who are taking CELMoDs need to worry about? And are those side effects similar to what you might see with like Revlimid or Pomalyst?
No, I would actually say that the CELMoDs are much better tolerated than the IMiDs overall. I'll give you a real life example. We had a patient who was on a clinical trial. He got a BCMA CAR T, and then he received iberdomide maintenance after, or he was supposed to. He took the medication. Unfortunately, the neutropenia was a major side effect, so much to the point that he did have to come off. But at no point while he was on the medication did he complain of anything that you would traditionally see with an IMiD like Revlimid, like he wasn't having any diarrhea. His fatigue was no worse than it had been while he was going through the CAR T. He didn't have a rash, for example. So overall, I think that the CELMoD category is much better tolerated than the, pomalidomide, lenalidomide, things like that.
Oh, good. That's like good news for patients who have to put up with those sometimes for years for maintenance therapy.
I mean, he was actually wanting to stay on the drug. He's like, why can't I stay on? And I'm like, well, you don't have anything to fight infection. I was like, that's a more serious problem potentially for you. So.
Okay.
Anything to add, Dr. Berdeja?
Yeah, no, I agree. But I was just going to say that notice how I said they may be better tolerated, whereas Nick said they're definitely better tolerated.
So there you go. Okay.
Well, I see all the follow-up maintenance folks on it. There's always something. So,
well, Mary makes me go and travel and go to meetings. Nick has to see my patients. So
exactly.
So he has much more hands-on experience.
That's not true.
No, I really do think that the CELMoDs are going to significantly affect how we treat myeloma, and it's going to be more of a replacement, because I think, in my mind, we still have the PIs and the IMiDs / CELMoDs are still the two big pillars, And then you're going to be, and then now with the anti-CD38 antibody, those were the three big targets. I do think that the BCMA and GPRC5D and all these other classes of drugs either will be complementary or will be sort of next in line for the foreseeable future, but potentially even frontline for especially for the high-risk population. So I think, having these sort of kind of making each class of drugs sort of the best, I think is important.
Mm-hmm. Agreed. Okay, so we're getting closer to the top of the hour, so I have one more question. So the final question for both of you is, was there any data presented at the IMS meeting overall that will immediately affect how you manage your patients? And what is the take-home message that you see from the meeting for patients? So Nick, I'll start with you.
I will keep it very brief because I'll leave it to the expert to kind of give all the details. But I think that the data that kind of most stood out, and Dr. Berdeja kind of touched on it earlier, was, having the CAR T prior to doing bispecifics and not losing that efficacy by having to eventually transition to the bispecific and still getting a response. The whole thing with myeloma obviously is sequencing, especially patients that have multiple lines of therapy or are high risk. And so I think that was the major takeaway looking at the data presented, is that, you can still have benefits post-CAR T by using other therapies such as a bispecific.
That's a good takeaway. Dr. Berdeja, your thoughts?
Yeah, no, I thought it was an excellent meeting. I think we were all a little bit sort of disheartened by the Canova results, which
I think so, right? I think we all were.
Is the Venetoclax dex versus pomalidomide dex. But I think, having said that, I think when you actually look at the data, you still see how much more powerful that drug is for that subset of patients. And like we discussed a little bit before, the more sort of optimistic take on this is that the Kyprolis, Venetoclax in combination with Kyprolis, looks fantastic, and at least in this early reports, looks just as powerful as any three-drug combination we've had in relapse refractory. So I still think there's life for this drug, and I think in the subset of patients, it just needs to be dosed appropriately. It should be looked at, and it is being looked at in clinical trials, sort of as a maintenance drug in this population, for example, and again in combinations. But I have to agree with Nick. I think the other thing that at least I learned the most about was the data coming out from the German groups and from Dr. Bayless's labs in Canada, looking at sort of mechanisms of resistance between CAR Ts and bispecifics, which is really helping us sort of determine how to best sequence these drugs. And so I think that is fantastic work that is ongoing there, and really is going to translate to optimal care for the patient with myeloma.
Yeah, agree. So the IMS meeting, I think, is getting better and better every year, actually. And it's actually good now that they're doing it every year instead of doing it every other year. And we all get to go to great places like Greece, and Rio. So that cannot be bad. It can't be bad. So and you get to
we all have to sacrifice for our patients.
I know. We have to travel across the world to go to the meeting, and but we learn all these amazing things, right? Which really helps the patients.
It's also a great meeting because it's not just about the data presented, but you bring the entire myeloma community together in a setting, in a forum where we're not being pulled in all different directions, right?
Right. So it's highly collaborative, and there's a lot of opportunity for people to collaborate on different topics, and talk about patients and sequencing and all these different things, right? So it's a great learning opportunity, and I thought there was more new data that came out from IMS this year than I've seen in some of the previous meetings, which I thought was great.
Right, and there's actually, Mary, there's actually a push to try and encourage people to present more data. Traditionally everything had to be presented at ASH. Right. Now we're learning, we start seeing more at ASCO. But the truth is, there's no reason to sort of kind of pile everything at once. Now we have this meeting sort of in between the two, the AMS, that if it's a myeloma specific data, all the myeloma eyes are in this meeting, and so hopefully that will translate to continuation. I think that's why you're seeing this, and we're hoping that it will translate to even further and more novel data that will be presented at this meeting.
Yeah, which is always great. So on behalf of the MMRF, I'd like to thank Dr. Berdeja and Nick Barkemeyer for joining me today. I'd also like to thank everyone for taking time out of your day to watch this session. And finally, I'd like to thank our sponsors, Adaptive Biotechnologies, Baxter, BMS, Cure, GSK, Janssen, Karyopharm, Regeneron, Sanofi, and Takeda Oncology. If you have any additional questions about what you heard in today's session, please don't hesitate to contact our MMRF Patient Navigation Center. Their phone number is 1-888-841-6673. Thank you and enjoy the rest of your day.